My name is Mirna, and I am a PhD student at Florida International University. And today it's my first time being an interviewer, and I'm so excited because I'm with Dr. John Butler. And the first time ISHI crew told me, "Are you interested in interviewing anybody?" the first person came to my mind was Dr. Butler. The first time actually I had an encounter with him, it was back in 2016, and it was when I first got introduced to forensic DNA typing. And I had a question in one of the pages in his book. By the way, his books are awesome, and we're gonna talk about it in a minute. And then I emailed him, but I thought that who am I for him to respond to me on my email asking for a clarification? So I forgot about it, and just 2 weeks after that, I got an email back saying, "Hey, sorry, I was in a conference, but this is my explanation." And I was so happy, and I was like, "Oh God," he responded, and it was just, for me, it's like my first time actually sending an email for an author and getting a reply. So I was like, okay, that's awesome. So with no further ado, I'm so glad that you accepted my invitation for an interview with you.
I'm delighted.
And I think in the forensic field, you are a leader in forensic DNA typing. And I am one of the big fans of your books and your knowledge, and I've learned a lot. I'm just curious to know, what was your trigger to actually write those books that a lot of students and practitioners are actually using?
So I grew up in a home with books. My father and my mother were both teachers. My dad was a college professor, and he's written about 12 books on farrier science, on how to make horseshoes and put them on horses' feet. And so since I was a kid, my dad wrote books. I'm supposed to write books eventually. I didn't know what the book was going to be on. I like history quite a bit, but I've always loved science, and so I had 4 years of biology in high school, and so got interested in forensic science when I learned about that as a profession. But this is before CSI became popular or anything. Yeah. And then just started learning about DNA and started working in it, and then realized there's lots of information that I've gathered as part of my PhD dissertation. So I built a website that was the star-based website. Yeah. And then I wanted to write those things down and make them available to kind of help teach people step by step how to do the process. And so that's when the first books were. Now it's been 20 years since those books were started, and I've done 5 so far. So.
That's awesome. I can say, like, when I read through them, it's just like the workflow or the way you wrote them is so easy to grasp and so easy to understand. You go to the table of content, you need a specific information, you just find it. Like, I just find everything I need in those books.
So there's a reason of that. It's called pedagogy, the process of thinking about how to teach something and the process of helping people learn. And so I spent a lot of time with a whiteboard thinking about each step of the process and how to design the teaching for that. And so each chapter is thought through many, many times, readjusting things to the right order. And then my wife, who's not a scientist, reads what I write, and so if she can't understand it, I have to rewrite it. So she helps make it understandable. So you can thank her for that.
Well, so. That definitely a big thanks then to her as well. That's really so inspiring, and I'm sure that's not only my opinion, but also a lot of other people who are using your books have the exact same, if not more, on your books. As a student, my interview might focus a little bit on some advice for new people in the field and new scientists who are really interested in forensics. Based on your experience, you know, there are, if we give an example about the United States or other countries, there are programs, especially we're talking here graduate level, that offer either degrees just as forensic science, or others that are STEM, like biology or chemistry or biochemistry, but then they have the forensics as a track. Do you see any difference between the two, or like any one that is better than the other?
It depends on 2 things. One's the professor, and the next is the curriculum. So who's teaching it and what's being taught. So I think you can have, there's 3 PhD programs right now, one at FIU, one at Sam Houston State, and one at West Virginia University in forensic science. And you can get biology tracks or toxicology tracks and stuff. But I think in general, it's typically if you get a degree in analytical chemistry or molecular biology, you're probably better off right now until those programs are developed further, but it depends again on the professor. And so if you have a good professor to work with, I know you do because he helped teach me many years ago, so that helps then I think you can be a very effective program in any school.
Okay, that's interesting. Yeah. So to be on the same flow here, we're talking about PhD programs, but there are also countries that are not as developed as the United States. And a lot of them are now entering the DNA field. And if we compare what we're doing right now here of rapid DNA, genealogy, it's way, microbiome, we will talk about it in a little bit. But what is an advice for you, you can give them to accelerate maybe and to be on the same level as other labs?
I think part of it is what question you're trying to answer, and so if it's to address sexual assault cases in your country or a homicide, to focus on the simple things to begin with, and not try to think I need to have all the tools in place that it happens in the United States or in the UK or somewhere else, and just realize that it takes a lot of work to get there. So by the small and simple things, you can get to the big things eventually. But I would say focus on getting started and just the process of handling samples, for example, and be able to extract DNA effectively, and not worry about having the fanciest equipment to begin with at first until you can build up the expertise there. So I've had a chance to visit almost 30 countries and visit and see labs in many of these countries, and see some are very well funded and some are not. It's very dependent on the country and on the individual laboratory. So
that's interesting. You visited Lebanon, or you didn't get
Not yet.
Maybe we invited when
yes,
I worked
I need
there.
to do that. So, yeah. I've corresponded with a number of people from Lebanon, just answering email questions and stuff. So.
As usual, like your humbleness and your help for everyone is just...
I enjoy helping other people. I'm trying to live up to my name. See, I'm a butler, so I have to serve other people.
Yeah,
right?
exactly. That's a smart one. That's a good one. So now a little bit into some technical science question in here. Can you envision a system like the beads cleanup or the magnetic beads that can substitute or delete the need of quantifying DNA for DNA typing?
So it depends on the type of sample, because if you have something where you expect to have lots of bacterial DNA, and if the beads could tell the difference between human DNA and bacterial DNA, that would be fine. You just hybridize to the human DNA, but usually it's capturing DNA in general, and so you're going to have a hard time separating whether it's plant DNA or microbial DNA away from the human DNA. But for some samples, I think it could be quite effective, just doing a quantitation, bead-based quantitation.
When you're expecting not a lot of bacteria in a specific type of sample.
Yep, it would depend a lot on the sample type.
Gotcha. That's interesting. So we've seen in the last 2 days in ISHI conference that a lot of nowadays focus in forensic DNA is going towards other subdivisions than DNA typing, such as DNA phenotyping, microbiome, proteomics. I would like to know your opinion about each one of those three mainly, and whether you see there is any possibility, or where you see them, how far they are from being admissible or used in actual casework.
Again, it depends on the question that's being asked and the types of samples that are being processed. So there may be some situations where the microbiome can be very useful, and it's usually, I think, it'll serve more as a supplemental to the core work that's being done with STR typing and so on, because of the databases. Everything comes back to how do you match it out to somebody else or connect it to other potential people, and that's where the databases come in. So if you don't have something already in a database, it can provide an investigative lead and assist in some things, but it's probably not going to provide the full solution. Nothing's going to provide a full solution. So whether it's the proteomics, or the phenomics, or whatever, looking at the phenotype of something, or the microbiomics, it's, you're always going to have some. It'll help in some aspects, but it's not going to provide the complete solution to anything. So.
Because again, it's not confirmatory as the STRs, for example, to compare and do some statistical, but there might be, as you said, investigative lead.
Correct. They can help with, and that's what the genealogy whole area is helped with. That is providing a new avenue to access samples that you couldn't, or people that you couldn't access previously. So that's where the microbiome can do the same kind of thing. It could answer a question from a different angle, and that can be very useful.
Yeah,
so.
That's interesting. And that's a question that backs up what probably you were saying. Do you think that proteomics or any other discipline might just replace DNA typing at some point in the future, or?
Again, you have to have something to compare it to, because always in forensics, it's always a Q versus a K, a question and a known. And if you don't have knowns to compare to, so it takes time to build that a database of knowns, if you will, to make that comparison. So if proteomics becomes more prevalent, and that could certainly happen as more and more research occurs there and it gets implemented, you still have the challenge of what are you going to be able to compare it to, and that will take time to build up. So I think time will tell.
Interesting. Yeah, probably also financially, whether they can build this big database as the CODIS nowadays that has
so you have the expense of the instrument. So in research, often you don't think about the practicality. It's just, can we do this? And so it has a different, academic research has a different purpose, which is, can we publish a paper? Can we learn something new from this? Can we extend the field? But at the end of the day, in the forensic lab, you have to make a practical decision. What can we do to actually help solve this case and help assist there?
That's very critical, yes. Thinking like business-wise and or like how you can implement it. Is it practical to implement it for, that's true. What, in the past 10 to 20 years, what you've seen as the biggest breakthrough after like the PCR and that breakthrough by itself in the field of forensic DNA typing?
So I'd say the biggest thing is the standardization, the fact that you have core loci, and that's what enables then the databases to be built and everything else to come around that. So it's not really an innovation in that way, but it enables everything else. So you don't, there's always cool things with new software, new tools, new ways to sequence the DNA. Those are wonderful and they extend the capability, but at the end of the day, can you help solve more crimes and connect more things? And that's going to be having lots of knowns to compare to is what enables that to happen.
Yeah, and I can say like the work that's been done for creating and maintaining those standards through NIST and other organizations is just very important. Providing labs who are trying to do validation and some controls and some known samples so you can verify and do accreditation for those labs. I think, as you mentioned, is very critical point, because you cannot trust any results if you're not sure that they're doing the right protocol.
You need data to support conclusions. That's the bottom line.
Exactly. Exactly. And what is this, like the strangest DNA profile you've ever seen? Since I'm pretty sure you've seen so many DNA profiles.
Well, working with the Starbase website, we take in duplicated alleles, triplicated alleles, triallelic patterns, and so on. Those are fascinating to see, and those are chimeric. Sometimes seeing chimeric profiles where you see mixtures from two different individuals that are part of one individual that happens. That's fairly rare, but those do occur. But probably the most interesting one was we saw a very large, it was a 29 allele at the D5S818 locus, and it was an insertion after it was sequenced. There was a big insertion that occurred. So it was just a very unusual insertion that occurred right next to the repeat, and so the primers made it very large. And so it's just 29 alleles way outside the allelic range that you normally would expect for that locus. So that would be one of the more unusual ones, I would say. That's
interesting. You've sequenced the sample to be able to know that there was this
it was a 48 base pair insertion that occurred. So Margaret Klein at NIST sequenced the sample when we found it, and so it was very interesting to see. So people for a while would send us samples that we would sequence and just kind of see these oddball situations, what's going on. Sometimes you see a primer binding site mutation. Those are fascinating to see. Why doesn't the primer bind properly? Because the sequence has changed relative to the normal templates. So.
That's interesting. Yeah, I can say actually when during my master's, I did collect some Lebanese samples, and we did do the conventional STR on them with NIST, with Dr. Pete and Dr. Sarah, and we did see tri-alleles in autosomal STRs and bi-alleles in Y STRs as well. Not conventional, like on the database, they were not found yet, or no one yet has observed. So yeah, those are unique and interesting to sequence, I think.
And it's fun to be able to share those with others. So that's when people would submit them to Starbase, when we put them up there. There's hundreds of those that are available now, and so having a catalog of that, so that anybody can go and look and see, oh, someone else has seen that same duplication or that triallelic pattern or whatever. So
yeah, I think, is it true that nowadays they actually, if they want to do the forensic compute the forensic parameters, they remove those from the
Typically that's
calculation?
done because it's not, we don't understand the mechanisms by which there are really copy number variants, so that an extra section of the DNA gets inserted in there. But how often does that happen, and how do you compute that from a genetic statistical standpoint? Usually it's just left out of the calculations.
But it gives more uniqueness for the
Correct,
profile.
yes.
So as a forensic application, I think it's like more unique. We can catch criminals more often that way.
Well, they don't occur all the time.
Yeah, of course, that's so true. So now a fun question. Can you juggle and do a rope trick at the same time?
So I've learned how to spin ropes and stuff, and I've shown that at meetings in the past. And I also learned how to juggle when I was in college and stuff. So, but I don't normally do those at the same time. So, and I'm not going to try to demonstrate that here today. So
maybe some practice, and then
yeah, maybe that's something I'll try someday.
And
but
another ISHI conference.
There you go. Yeah. It's fun just to attempt those things sometimes. Just to people don't expect that from me, I think. So to spin a rope or to juggle. So.
I mean, that's not easy. I can see.
Just take some practice. That's all. So.
I barely know how to play ping pong, but other than that, I don't know. Awesome. We know that you are going to be the president for ISFG, and it's going to be held in Washington, D.C.
Yes, in 2 years. Yeah.
Yeah, that's exciting, and I'm looking forward for that one. Are you planning as a president, since in D.C. and president, to go to the White House?
I don't know if that'll happen. In terms of, for me personally, I've been to the White House before,
nice.
but not in this capacity. I probably wouldn't. But as the president of ISFG, I'll have a chance to kind of welcome people to come to the United States, which will be exciting. There's 84 members, or 84 countries that are part of ISFG right now, so it's exciting to see. My goal is to learn how to say thank you in many of their languages. That would be kind of fun to be able to greet people and to make those connections. But I think, to me, the neatest thing is ISFG is a chance for a scientific community to come together. And I'm excited to see people come to the United States, and also for the United States scientists that normally don't get to go overseas, to be able to see some great science. And so I think it'll be fun to see that in a couple of years.
That's going to be really interesting. I can teach you the Lebanese way of saying.
Okay, I would love that.
Yes, I'm saying Lebanese, not Arabic, because each of the MENA region has different dialects. So yeah, that would be interesting. I would be looking forward definitely for this meeting as well, and I'm pretty sure it's going to be just perfect. I think I would sit here for hours just speaking with you, because every time I talk to you, I learn a lot. But I think I would like to make it a little bit shorter this time. And last funny question, I think, is if aliens exist and they come to here, do you think they would have the same DNA as we do? And if not, what kind of DNA typing or what kind of method we need to identify those?
Well, first you'd have to swab them and get a DNA profile from them, right? So, but how would, my philosophy is, if the primer doesn't bind, a DNA profile you will not find. So there you go. So it's just like, so it'll depend on whether or not the primers will bind to the sequence. And so we wouldn't know until we're able to sequence or at least attempt it. So as with anything in science, give it a try and see what happens. So.
Exactly. Just do an experiment and see.
Yes. So.
Awesome.
And if it fails, then you have a species identification. The species not specific to them, and you have a null allele because it's not, the primers aren't going to bind properly. So.
Yeah, if we want to speak science here.
Yeah.
Awesome. I really, really appreciate it. I cannot find the words to say thank you, I think. I would say shukran, which is thank you in Arabic, and I would be looking forward to meeting you again in other meetings and keep this connection. I really appreciate everything, and I don't know if you would like to say anything before.
Well, I think one of the things you learned is asking a question is critical to learning. And so I would say, just like you did when you first contacted me and asked me about the different types of errors that were being reported in the appendix in that book, and did I classify them correctly? And questioning that's a wonderful thing. So that's what a scientist does, is ask questions. And so if you're afraid to ask questions, then you're not able to learn further. So keep asking questions, and
Thank you.
and don't feel afraid to ever ask me a question. I'm happy to try my best to answer them.
Okay, I really appreciate it. That's for sure a very important advice for new researchers and new scientists in this field and in other fields as well. As you said, in the beginning, yeah, I would tell you I would be afraid to ask.
Sure,
but
people get intimidated, but I'm just a regular person, so ask the questions. So.
Yeah, I wouldn't want anyone to take like to think like wrong of like why would is it a dumb question, or like no, as you said, no question is dumb. You need to learn and understand. Yeah, thank you so much for your time, and thank you for watching.